So let's start with the first type of protein activity that we'll discuss, which is enzymatic catalysis. And we already mentioned that life is based on chemistry, that there are many, actually thousands of chemical processes or chemical reactions that occur within cells and living organisms, and which are required to drive life processes. These reactions do it by forming metabolic pathways, by transporting solutes across membranes, and in other ways as well. The problem is that in order to do that, these chemical reactions have to occur within fractions of a second, very fast. And in fact, many of these reactions are very slow. They require between minutes and billions of years to occur under room temperature conditions. And this means that living organisms had to find a way to accelerate their chemical reactions significantly in order to remain, to stay alive. Now, theoretically speaking, there are two ways to accelerate chemical reactions. One is to apply heat, which is not, as you can guess, not a viable option for living organisms because they can't tolerate high temperatures. The other one is to use a catalyst, which means to use a chemical species, a compound, that when it comes with contact with reactants, it accelerates their reaction to form a product. And this is indeed the solution that was adopted by living organisms. And in fact, there are many different compounds that could act as catalysts because many of them surround or they are present in the natural environment of living organisms. And some of them are even very simple, like metals or clays. But these simple catalysts were not adopted by evolution. The chemical species that were selected by evolution to act as catalysts were proteins. And there are several reasons for that. The first reason is that proteins are indeed very efficient in catalyzing chemical reactions. I'm referring, of course, to proteins that act as enzymes, as catalysts. They can accelerate chemical reactions by many orders of magnitude. But more importantly, proteins are specific. They each enzyme, each protein catalyst catalyzes only a specific type of reaction involving a specific type of substrate or reactant. And this is a huge advantage because when you have thousands of different chemical processes inside one cell, you need the catalyst to be specific towards some of them. Otherwise, it will accelerate all of them. Another advantage of protein catalysts or enzymes is that they can be controlled. The cell contains many small molecules that can bind to enzymes and change their activity. It can make their activity faster or slower, but this ability to control it is very important. And finally, an enzyme can couple spontaneous to non-spontaneous reactions or processes. In other words, while many of the reactions that occur within cells occur spontaneously, others cannot, and they require an external energy to occur. And what enzymes can do is to drive a reaction that requires energy by accelerating in parallel at the same time a reaction that gives away energy, that releases energy, and to channel the energy from the second process to the first one, and therefore driving it. This is another important advantage. I mentioned the specificity of enzymes towards the type of reactions that they catalyze. And indeed, if we look at the enzymes found so far in nature, we see that each of them can catalyze only 1 out of 6 types of possible chemical reactions. And we can see these types here and in the following slide. The first type, catalyzed by the largest group of enzymes, are called oxidation reduction reactions or redox reactions. And they involve the transfer of electrons from one molecule to another. For example, in this case, we have ethanol, an alcohol, and it gives away its electrons to another molecule called NAD. which converts ethanol into acetaldehyde. It oxidates it into acetaldehyde, and NAD is reduced to NADH. The second type are reactions that involve the transfer of a group, a chemical group, from one molecule to another. In this case, the amino group of alanine, which is an amino acid, this group is transferred to this molecule, oxoglutarate, converting it into glutamate, which is an amino acid, and alanine, when it loses the amino group, becomes pyruvate. The third type are hydrolytic reactions, reactions that involve breaking up bonds inside a molecule, and they do this by using water molecules that act as nucleophiles. So in this case, the water molecule attacks the aspirin molecule, specifically the ester bond inside it, and converts it into salicylate and acetate. The fourth type are reactions that involved also the breaking of bonds inside molecules, but not by using water, but by using chemical groups inside the enzyme. Moving on, the fifth type are isomerization reactions. These are reactions that convert one chemical isomer of a molecule into another one. For example, in this case, the reaction converts this isomer of the double bond in the molecule into another isomer, from trans to cis. And finally, we have ligation reactions, which takes two molecules and connects them into one. What happens is that these reactions create a chemical bond between the two molecules. So here we have two alanines, two amino acids, and they react with one another in a ligation reaction to form a peptide, a dipeptide of two alanine amino acids. So how do enzymes accelerate chemical reactions? To understand that, we must first understand how a chemical reaction occurs, or more specifically, how the energy of the system changes throughout the reaction. So let's look at a hypothetical reaction that is shown here, in which the reactant A is converted or broken down into the products B and C. For this reaction to occur, A must first go through a transition state called A tag, which has high energy. So in other words, in order for A to become B and C, it must first gain this amount of energy, become A tag, and from there, the reaction can be completed easily. This energy is called the activation energy, or EA, and this is what limits the rate of the chemical reaction. The higher this energy is, the slower the reaction. And we can see that in the Arrhenius equation over here, where K is the reaction rate, A is a prefactor, which is obtained empirically, and R is the gas constant, and T is the temperature. And we can see that when EA, the activation energy, is very large, this expression is very small. And therefore, K is very small, and the reaction is slow. So what enzymes do is that they accelerate the reaction by lowering the activation energy. They bind, each enzyme binds to its substrate, in this case, the reactant A, and stabilize or reduce the energy of its transition state. When the transition state becomes more stabilized, it has less energy, it becomes easier to reach it and to complete the reaction. So how do enzymes stabilize the transition state? They do it by using a variety of mechanisms, and we won't discuss them in detail. You can find this discussion in chapter 9 of the book. But we'll just say that they use very different mechanisms, and often the same enzymes uses a combination of different mechanisms. So, for example, some of the enzymes use electrostatic stabilizations. They have amino acids that interact with the transition state and stabilize it. Others use acid-base catalysis, which means that they donate protons or abstract protons from the reactant, and this makes it easier for the reactant to reach the transition state. Others use metals or even covalently attach to the reactant, but still very transiently. And all of these mechanisms, they have the net result of, again, of stabilizing the transition state, reducing its energy, which allows the reactant to go more easily towards the products.